Hello and welcome back, everyone. For the next company webcast, we welcome Genvara BioPharma, a privately held company. Joining us from the company is Tom Cure, the company's Chief Executive Officer, and Dr. Michael Bristow, the company's Chief Science and Medical Officer. The team will be taking us through their slide presentation, and if time permits, we'll have a brief Q&A session. So with that, Tom, Michael, I'll go ahead and turn the floor over to you to begin.
Great. Thanks a lot, Robert. My name is Tom Coyer. I'm President and CEO of Genvara BioPharma, and with me here today is Dr. Michael Bristow. He's our Chief Science and Medical Officer and also the founder of Genvara. And we really appreciate the opportunity today to talk to you about our late-stage cardiovascular development program. So we're a company based in Colorado, and we're sitting here in our offices near Boulder, looking out at the Colorado Front Range. And I wish I could turn the camera around because I'd argue we have one of the best views in biotech. And any of you who want to come out and visit us are more than welcome to confirm that. So Genvara is focused on the therapeutic application of genetic variation and the development of precision medicines to treat cardiovascular disease. Here are the key investment highlights for the company. We're very unique in that we're a private company with a late-stage cardiovascular asset that's poised and ready for a phase 3 clinical trial. Our lead asset, Gencaro, is a genetically targeted therapy that's being developed for the prevention of atrial fibrillation in heart failure patients. And this is an unmet medical need because there are currently no effective drug therapies for this condition. This also represents a very large market potential in the range of $1 billion or more in the US alone. And we also have recent scientific discoveries that should extend the patent coverage to 2044. 2 pharmacogenetic clinical trials have been conducted with Gencaro, and the data from these trials informs and de-risks our clinical program. We've also interacted with FDA throughout the trial design process, and thereby we believe we've reduced the regulatory risk. So Gencaro is one small pivotal trial away from FDA approval, and our design is for 350 total patients, which in the cardiovascular world is a very small, efficient, and cost-effective pivotal trial. We're currently seeking corporate partners and investors to help finance this pivotal trial, and $65 million will fund the trial costs and the company operations to get us to an FDA submission for approval. And while Genvara is a new company, we already have the key leadership in place with the right expertise and specific experience to execute our plan. So we're in the business of developing therapies to treat cardiovascular disease, and one of the primary reasons is because this is a leading cause of death and morbidity in the world, and both AF and heart failure are major contributors to this. For heart failure, upon diagnosis, the overall 5-year survival rate is only around 50%. Now, heart failure is classified by LVEF, or left ventricular ejection fraction. And this is basically a measure of how well your heart is pumping blood out to the body. A healthy heart's LVEF is between 55 and 70%. And heart failure classifications by LVEF are listed on the slide, but I want to focus on the one that's highlighted here. And that's an expanded group of patients with mildly reduced preserved LVEFs in the range of 40 to 54%. And this is known as HEP-MPREF, and I'll come back to this in a few minutes. Atrial fibrillation is also a very serious condition, particularly in combination with heart failure, and it increases the mortality, morbidity, and stroke risk in those patients. Unfortunately, despite decades of pharmaceutical development to address both AF and heart failure, the prevalence of both is continuing to increase at a rapid rate. Also, one condition can trigger and or worsen the other. And there are currently almost 7 million patients in the US with heart failure and about 60 million worldwide. And about 45% of those patients with heart failure have atrial fibrillation. So we're talking about very large patient numbers here. So it's clear there's a need for better solutions to address both AF and heart failure. And there are currently no approved effective drug therapies for this condition. Beta blockers on the left here are approved for a treatment of reduced ejection fraction heart failure, or HFrEF, where the LVEF is below 35%. But they are not indicated for AF prevention, nor are they indicated or effective or approved for higher LVEF heart failure above 35%. Antiarrhythmic drugs here in the middle used to treat AF outside of heart failure are not durably effective and are not safe in heart failure patients, and as a result, several are contraindicated. Ablation is a surgical procedure that's effective to treat AF and heart failure, but it's invasive, it's very expensive, and often requires repeat procedures. So our solution to this unmet need is Genvara, which has the potential to become the first pharmacogenetically targeted precision medicine therapy to treat cardiovascular disease. This is a drug that's ready for a phase 3 trial that's been designed on the basis of results from 2 earlier pharmacogenetic trials with Genvara and with substantial feedback from the FDA. So we believe it's significantly de-risked and has a good probability of success. And when approved, Genvara will also have a very unique label that will support its commercial differentiation. So what is Genvara? It's a small molecule oral beta blocker that has a unique and differentiated mechanism of action. It works through biological and disease-modifying action by reversing the structural and electrical remodeling of the heart that occurs during heart failure. This is known as reverse cardiac remodeling, and it's a very important feature, as I'll explain in a few minutes. It's the only beta blocker that's shown effectiveness for genetic targeting, and up to 65% of the overall patient population has the favorable genetic profile in terms of efficacy. So this isn't a 10 or 20% subgroup that you might typically see for a personalized oncology therapy. Instead, Genvara has the potential of benefiting a very large percentage of patients. Genvara's mechanism of action is clearly differentiated from other treatments, and there are extensive data from clinical trials in over 3,700 heart failure patients, so the safety profile is favorable and well understood. So let's dig a little bit deeper into the data from the 2 prior pharmacogenetic trials that I mentioned earlier. So on the left is the BEST trial, where that was a very large heart failure study looking at Genvara against placebo. And there was a large DNA sub-study conducted as part of this trial. For the 441 patients that had the favorable genotype, which is the beta-1 receptor 389 RGARG genotype, and this is present in about 50% of the overall population. For those patients, a 74% reduction in AF onset was demonstrated against placebo, and that's shown in the Kaplan-Meier curve here on the left. On the right here was the genetic AF trial. This was a 267-patient phase 2b trial studying Genvara against an active beta blocker comparator, which was long-acting metoprolol. And it was also targeted to the same beta-1 389 RGARG genotype in a predefined population of patients, predefined in the statistical analysis plan, which represented about 75% of the patients in the study. There was a 46% reduction in AF recurrence with Genvara. And this is going to be our target population for phase 3. We call this the precision phenotyping cohort. It's described in the highlighted section toward the bottom right here. And basically, this eliminates patients with long-standing disease. And by long-standing, I mean patients who've had either AF or heart failure for more than 10 years, or their AF diagnosis preceded heart failure by more than 2 years. Patients with long-standing disease have been extensively treated and unfortunately are generally non-responsive to new therapies, particularly in the early stages of dosing when you're transitioning a patient off of a therapy they've been taking for multiple years and onto the study drug. Related to that, there's another important feature and finding from genetic AF that I need to explain here. So the endpoint for this study was time to first event, or TTE, which means the time to the first recurrence of a symptomatic AF event in these patients. This is the endpoint that FDA has traditionally used for approval of AF therapies. The problem with that for Genvara, as I mentioned before, it works through reverse cardiac remodeling, and it takes time for this disease-modifying mechanism to take place. As you can see here on the circled section on the right graph, many of the AF endpoints, the TTE endpoints, almost half occurred in the first month before the drug really had a chance to work. After the first month, there's a clear separation between bucindolol and metoprolol across the treatment period, which in the case of this trial was 6 months. And overall, there was a 46% reduction in AF occurrence for this cohort of patients. But these data clearly demonstrate why TT is not the best endpoint for this drug with a mechanism like Genvara, because you need an endpoint that gets you through this early period of instability and evaluates the performance over the entire treatment period, and that gives the drug time to work. One other important thing we learned from the genetic AF trial was that there was a higher incidence of AF in patients with mildly reduced to preserved LVEFs, specifically between 40 and 54%. And this is the HEP-MPREF group that I mentioned earlier. And also the effectiveness of Genvara for AF prevention was highest in the same group. So to reiterate the three key learnings from the trial: don't enroll patients with longstanding disease. There's a need for a better endpoint to evaluate the effectiveness over the entire treatment period, and target patients with LVEFs in the range of 40 to 54%. Now, this slide, also included in the genetic AF trial, was a sub-study of patients using implanted devices to continuously monitor heart rhythm. And by using these devices, you can calculate what's called AF burden, which is essentially the percentage of time during the day that a patient's in AF. From these data, you can see what I was just talking about. You see here in the first month on the left, there's not much difference in AF burden between Genvara and metoprolol, but over time, there's a clear separation, and this gets more favorable over the treatment period. Across this whole treatment period, there's an overall 36% reduction in AF burden, and at the end of the dosing here at 6 months, there was a 55% reduction with Genvara, so the efficacy gets better over time. From the data I just showed you, you might conclude that AF burden would be a good replacement as a primary endpoint for time to event, and we considered this as well. However, in discussions with FDA, it became clear that reduction of AF is not enough. The FDA also wants an endpoint that demonstrates that the patient feels better, that the symptoms are reduced, because not all AF is symptomatic. So we needed to come up with a more appropriate endpoint that evaluates the symptoms of the patient, but also studies the effectiveness of the drug over the entire treatment period. Through extensive discussions with FDA, we developed a novel endpoint called the symptom burden of AF, which we believe addresses these needs. This endpoint is based on a questionnaire tool that allows a patient to report their symptoms, from which is calculated a symptom score. We actually utilized a version of this tool in the genetic AF trial, as the data here on the slide indicates. So what these two graphs show is the comparison of the traditional time to event, or TTT, endpoint on the left graph, with continuous tracking of symptom burden here on the right graph. And these are data for the same group of patients in the trial, which represents our target patient population for the phase 3 trial design. The important point of this is, while the TTT endpoint does show a reduction in AF for Genvara versus metoprolol, there's quite a bit of variability in the data, largely caused by instability over the first month of treatment, and overall the p-value is not significant. For the symptom burden endpoint on the right, there's a p-value of 0.003, which is statistically significant, and the data are much tighter. And there's a similar reduction in symptomatic AF of 44%. So after reviewing these data with FDA in detail, they've agreed with symptom burden of AF as the primary endpoint for use in our phase 3 trial, which we believe is a really good and important outcome. So this is a summary of the endpoints that we monitored in the genetic AF trial, and I want to focus on the bottom half of this because we've already discussed the top half. So there were a number of additional endpoints measured for the entire study population, including patients with longstanding disease, that show that Genvara is more effective than metoprolol. This includes ECGs and normal sinus rhythm, where there was an improvement of almost 40% with Genvara. There was also a reduction in other AF interventions, such as ablations, electrocardioversions, and administration of antiarrhythmic drugs, a reduction of 32% for Genvara. Also, there was a twofold reduction in a common heart failure biomarker, proBNP, and a 61% reduced incidence of bradycardia. All of these endpoints that I mentioned have p-values that are significant, and again, were for the full study population, and they clearly demonstrate that Genvara is a better drug than the active comparator metoprolol for prevention of AF in these patients. And of course, we'll be monitoring all of these endpoints in our phase 3 study as well. Okay, here's something new and exciting that really adds to the pharmacogenetic benefit of this drug. Recently, we discovered that Genvara is unique among beta blockers in that it internalizes beta receptor haplotypes variants that are normally non-internalizing, and this results in cardioprotective signaling. So what is a haplotype? It's a set of DNA variations or polymorphisms that are inherited together and that are located on the same chromosome. And so what does all this mean? Well, it means an improvement in the favorable target genetic profile from 50% of the population, which was for the beta-1 R-charged genotype, to around 65%, with an associated 30% increase in the patients that can benefit and in the potential market. It also means that efficacy is improved versus standard beta blockers, and there's even a super responder population representing 25% of the patients with an over 60% reduction in AF versus other beta blockers. This discovery has also allowed us to file additional IP, which extends our patent coverage to 2044. And this haplotype finding has been corroborated via data analyses from multiple clinical trials. It's also been verified in laboratory experiments using human model systems. There's a lot of really interesting data behind this, and we plan to publish that and also study these haplotypes in our upcoming phase 3 trial. So now I've described the data that have informed and de-risked our phase 3 trial, which we call Precision AF, and here are the key points. We now have an enriched and responsive patient population, which eliminates the non-responsive patients with longstanding disease. We have a much improved primary endpoint that meets FDA's requirements for monitoring symptoms and that allows the drug the time to work by evaluating its effectiveness over the entire dosing regimen. And this endpoint has been reviewed extensively with FDA and now has been described in detail in a recent publication. We're focused on mid-range LVEFs above 40%, where AF is more prevalent, where the unmet need is the greatest, and where the highest efficacy was observed in the phase 2b program. And finally, this trial incorporates the pharmacogenetic targeting of haplotypes, which expands the favorable responsive patient population from 50 to 65%. All of this results in a very efficient pivotal trial design, particularly in the cardiovascular world, where phase 3 clinical trials typically involve thousands of patients to demonstrate an efficacy benefit. This is a 350 patient trial that will have about 100 clinical sites. We estimate the trial cost of around $50 million with a timeline to top line data about 27 months from the first patient in. There's also a big upside of this trial successful because we believe we also have a good chance of obtaining labeling for low ejection fraction heart failure, or HFrEF, which would significantly increase the patient population that would benefit and would also expand the market. So speaking of the market, we believe based on a primary research study conducted on Genvara by Syneos, that the market potential for this drug is around $1 billion annually in the US alone. There's also a robust opportunity in international markets, and as I mentioned, a good chance of expanded labeling beyond what was researched in this study by Syneos. Also, recent acquisitions in the cardiovascular space, as outlined on this slide, support a high exit potential for Genvara, with all these examples having valuations of $1 billion or more. And this reflects the pharma industry's continuing focus on this space and the need for market-ready cardiovascular therapies. So our roadmap to this market and to this exit potential is clear. We're seeking partnerships and investments to get funding needed to run the company and to execute our pivotal clinical trial program, get us to top-line data and to a regulatory submission. To use a sports analogy, and since it's still football season, we're in the red zone. We need a final push to get us to the pay dirt. So that's our goal. That's why we're here at this conference describing what we believe is a very unique and interesting opportunity. Thanks very much for your attention. At this point, we'd be happy to address any questions.
Tom, thank you so much for that presentation there. Maybe just a couple of questions here in the time that we have remaining. You've explained that Genvara is targeted to patients with a favorable genetic profile. What type of testing is required to ensure the right patients are selected?
I'll let Dr. Bristow address that one.
Yeah, thank you. The first point is that we're measuring haplotypes for both beta 1 and beta 2 adrenergic receptors, not just beta 1 genotypes that we previously have targeted and that Tom was talking about. And that's because haplotypes are the genetic substrate from which a protein is made. If you measure genotypes only, you're measuring an admixture essentially of polymorphisms that are coming from both parents' chromosomes, and you cannot discern the precise haplotype structure. So it's a very easy thing to do. We do this test every day in our laboratory, essentially. So you start with 5 mLs of blood, you extract the DNA, you do the genotyping in the usual way, which is basically creating an amplicon around the polymorphism and then sequencing it. And then you basically, through a likelihood method of computer modeling, determine the haplotype. And that, because these polymorphisms in both the beta 1 and beta 2 receptor are in strong linkage disequilibrium, the accuracy of this method is greater than 99%. There's a small percentage of double heterozygotes that need to be long read sequenced later. But the point is, you can do this in the lab quite quickly with a turnaround time of 48 hours. So the plan for phase 3 essentially is to do the testing in our own laboratory, essentially, but at the same time, consider developing an FDA approvable diagnostic test. So we will be in discussions with CROs that develop diagnostic tests for that purpose.
Okay, excellent. Timelines, costs for clinical trials, often very difficult to predict. What gives you the confidence that you can execute on Precision AF, the pivotal trial here, to the plan that you've outlined in the presentation here?
Yeah, that's a really good question. It's one that those of us in clinical development get asked quite a bit, mostly by boards of directors. And you're right, most clinical trials take longer and cost more than initial estimates. What gives us a confidence in this case that we can meet or exceed our plan is that many of the same team members that are now at Genvara were responsible for managing and completing the genetic AF phase 2b trial that I talked about in the presentation. And the Precision AF pivotal trial is very similar in terms of design, in terms of the patient population and the scope. Genetic AF was a 267 patient trial, and Precision AF will be 350 patients. So similar in size. We know exactly what needs to be included, what sites to recruit, and what the estimated budget will be based on the prior experience. We also have a good handle on enrollment rates. We know which countries and sites will be the high enrollers, and we'll be selecting those accordingly.
Okay, great. And just maybe one last question, coming back to something that was discussed in the presentation, was the sort of that new recent scientific discovery regarding the beta receptor haplotypes. Maybe elaborate on what this discovery is, what it means in terms of Gencaro's effectiveness and really differentiation.
Yeah, so the discovery began with work we did in another non-Gencaro clinical trial looking at haplotypes in a study that was measuring genetic effects, or in our case, haplotype effects on ventricular fibrillation and acute MI. And what we found was that internalizing haplotypes of both the beta 1 and beta 2 receptor were protective essentially against ventricular fibrillation. That study's published. We then formulated the hypothesis, since we knew that bucindolol internalizes both beta 1 and beta 2 receptors through work we had done years ago. If bucindolol or Gencaro does this, what happens to the non-internalizing receptors when they get internalized? So we went back to the BEST trial DNA sub-study as well as the genetic AF trial, where we had haplotype determination, and essentially showed that if you have a non-internalizing beta 1 and beta 2 receptor, both of them together, you get this big boost in efficacy. So that's basically how the observation was initially made and how it was essentially confirmed. So we have IP, as Tom mentioned, that's submitted on this. We have an NIH grant sitting waiting for review. And we have a big publication we're about to submit as well on this. So the magnitude of effect here is about the same as with the beta-1 ARG original genotype that we were targeting. We essentially double that efficacy, if you will, and increase the population now that will have enhanced effects from Genvaro, both for atrial fibrillation as well as for heart failure endpoints.
All right, great. We've got maybe about 2 minutes left here. Any closing comments here or takeaways?
No, just thanks for your attention. We do have a lot of data around this haplotype discovery and around all of our clinical trials that we've executed. We'd love to share that with those folks out there that are interested. We could spend a day or more going through it all, but there is a really sound basis for what we're doing, and look forward to further discussions with some of the folks in the conference on that.
I would also add that everything you've seen today, with the exception of the haplotype data that I mentioned we're about to submit for publication, is published. If anyone wants to see these publications, contact us. We'll send you the links essentially to the papers.
Fantastic. Well, we'll go ahead and we'll leave it there. Thank you, gentlemen, so much for the presentation, and thank you to everyone for watching. If you do have any questions and I can help facilitate, please send me an email, Blum, B-L-U-M, at lythampartners.com. As a reminder, we have a few presentations, fireside chats still remaining here today, so please don't go anywhere. Again, gentlemen, thank you so much for the participation today. Really appreciate it.
Thank you.
Thank you.